Hello everyone, my name is Farhad Jahangiri. My presentation is about HBV Molecular Epidemiology. Welcome to my presentation. Hepatitis B virus is highly contagious and causes liver disease. Globally, more than 350 million people are chronically infected, and among them, about 80% are from developing countries like Bangladesh. Over 800,000 people die annually in the world. Resistance to existing drugs and vaccines are common phenomenon due to mutation in HBs antigen A determinant. Due to lack of about mutation and subtype of HBV in Bangladesh, this study strongly demanded to be documented. Genomic variation of HBV genotypes is determined by genomic relatedness, where the standard cutoff of divergence in about 8% in Bangladesh. The most prevalent genotype is genotype C among the chronic infection of HBV in on study, and genotype D among the patient with the chronic infection in a tertiary care hospital in another study. I used for my presentation from research a molecular epidemiology of hepatitis B virus isolated from Bangladesh. Here is the first page of my presentation research. Molecular methods. Sample selection. A total of 385 patients with jaundice-like illness were included in this study, and blood samples were collected from different medical college hospitals of Dakota City. None of the patients were HPV vaccinated. Serological analysis. Serum samples were subjected to analyze hepatitis B surface antigens by ELISA kits. DNA extraction. HPV DNA of several randomly selected HBs antigen positive samples was extracted from 200 microliter of plasma using pathogen kit. Extract DNA was stored at minus 20 centigrade until use. Polymerase chain reaction and DNA amplification. Partial S gene was targeted to be amplified in this study. A product of 491 base pair of partial S gene was amplified using primer pair as forward and reverse primers respectively, corresponding to nucleotide position 370 to 861. Post-PCR purification and sequence determination. The corresponding applicants were purified using the PCR cleanup system. Sequencing was done with the same primers used to amplify the 491 base pairs partial S gene sequence. Determination of genotypes and subtypes. After obtaining the sequences of partial S gene was performed to get the sequence similarly with other sequence deposited in the NCBI. The reference sequence for this alignment were located and foreign S gene sequence collected from NCBI Gene Bank. Results: Genotype D as most prevalent genotype in Bangladesh. A total of 54 samples were observed HBs antigen positive. Among them, 19 samples were randomly selected to be analyzed by PCR and then nucleotide sequencing. After phylogenetic analysis, 14 samples were found as genotype D, followed by genotype A, and then genotype C. Phylogenetic tree revealed the similarity of these sequences varying regionally across the world. Furthermore, genotype D was composed of two subtypes, namely AYW3 and AYW2, where subtypes ADW2 and ADR were specific for genotype A and genotype C, respectively, as shown in picture. Evolutionary relationship of HPV with other circulating HPVs. After constructing phylogenetic tree, observed a strong similarity of partial S protein sequence with other Bangladesh sequence up to 97%, and sequence from India up to 100%, and Malaysia 96%. However, sequence comparing genotype D were evaluationally diverted. On the other hand, there were several amino acid mutations. Mutations might have possible effect on structural and functional activity of S protein, which might be significant cause of resistance. Phylogenetic tree of EBA alignment revelate that our sequence were closely related with other Bangladesh exciting sequence with similarity of more than 90%. Phylogenetic analysis revealed the high similarity of genotype A, C, and D sequence with the sequence from India, Malaysia, and India respectively in the picture. Multiple mutation of sequence in A determined region. Several mutations observed in A determined region of S genes. These mutations probably were a strong impact of function of S genes, especially resistance of different drug as well as vaccine. Conclusions. This study is unique for three reasons. First, a detailed mutation profile of A determinant region of S gene was delineated. Second, amino acid compression sequence with different existing Bangladesh and foreign sequence was described. And lastly, third, this is the first comprehensive study detecting the mutation and subtype of HBV in Bangladesh. This study has importance to direct clinicians to diagnose the disease properly and decided to choose of drug or vaccine for the treatment of hepatitis B and HBV carriers. It seems like that combination therapy will become the new wave of the future in HPV therapy, but many questions remain to be resolved by clinical investigation. The past few years have seen the development of plethora of the new drug for the treatment of HBV infection. If, as expect, it proves to be superior, which combination of drugs should be used for initial treatments? Is combination therapy truly superior to monotherapy, clinical as well as virologically? These are the references I used throughout my presentation. Thank you for viewing my presentation. I hope you enjoy going through it.